Hello everyone, I shall discuss today an important central neurotransmitter, the gamma aminobutyric. This is an inhibitory neurotransmitter found in brain and spinal cord and sparingly in entire nervous system. It is amazing that GABA is mostly localized in the brain and central nervous system. In brain, it is concentrated in nigrostriatal circuits as interneurons and also found throughout the gray matter. About 20% of the brain interneurons are GABAergic and they are located in limbic system, basal ganglion, thalamus, reticular activating system. These are the areas which are involved in maintenance of trance, gait and consciousness, sleep, alert. The GABA is synthesized from the glutamate which is obtained from glutamate, and the enzyme needed are present in the brain neurons. You will see that GABA does not penetrate blood-brain barrier and therefore is synthesized de novo in the GABAergic neurons. There are specific enzymes which are present there. One important enzyme is GAD, that is glutamate dehydrogenase, and second is GABA transaminase. Now, sometimes we see that succinic semialdehyde is converted into GHB or sodium oxybate. Now, alpha-ketoglutarate is obtained from Krebs cycle and is transaminated to glutamate by a GABA transaminase GABA T enzyme. GABA is formed from glutamate by a glutamic acid decarboxylase, which is very important enzyme found in neurons synthesizing GABA. Interneuronal and astrocytic GABA is inactivated by this enzyme, that is transaminase, to succinic semialdehyde, and that gives glutamate in the presence of alpha ketoglutarate. Now this shunt is important in maintaining adequate levels of the GABA in brain to provide adequate inhibitory effect. Therefore, GABA transferase is both a synthesizing enzyme and for degradative enzyme. As a synthesizing enzyme, it provides glutamate, whereas it degrades GABA once it is formed, stored and utilized. An alternative minor pathway of GABA synthesis involves diamine oxidase and aldehyde dehydrogenase, which act on the precursor putrescine. GABA so synthesized is stored in presynaptic vesicles, and this storage is through a specific transporter called as vesicular GABA transporter or VGAT. In addition to this, the neuronal uptake of GABA is conducted through 4 transporters which play an important role in termination of the GABA action. GAT1 is an important neuronal recycling transporter for GABA, and it is mostly found in presynaptic neurons of neocortex, cerebellum, basal ganglion, brainstem, spinal cord, retina and olfactory bulb. And tiagabin, an anti-epileptic drug, inhibits this enzyme. GAT2 is found in choroidal plexus, meninges and peripherally in kidney and liver. Since it is present in choroidal plexus, that means it is involved in formation of CSF. GAT3 is found in glial cells, astrocytes, and is concerned with uptake of GABA in the astrocytes. There is another enzyme, SLC6A2 or BGT1, that is found at the extrasynaptic sites in the cortex and hippocampus. So this is where the GABA is synthesized, stored, released and taken up back. The typical mechanism that is seen in many of the monoamines in the brain and Now GABA acts through two types of the receptors, GABA A receptor and GABA B receptors. They are structurally and functionally dissimilar. For GABA A is a ionotropic receptor, whereas GABA B is a metabotropic receptor. GABA A receptor is located postsynaptically on the nerve membranes in the brain and spinal cord, and the surface of the membrane that is in contact with the synapse will have GABA A receptors, but they are also present at sites away from that contact. So they are called as extrasynaptic or extraneuronal receptors. They play important role in mediation of inhibitory actions of certain CNS depressants and general anesthetics. Now, GABA A is a ionotropic receptor, just like other ionotropic receptors, and pentameric in structure, flower-like pentamer consisting of 2 alpha-1 subunits, 2 beta-2 subunits, and 1 gamma-2 subunit. And these 5 subunits encircle the chloride channel through which chloride can enter into the cells. This is the most common pattern, and this is followed by variations like alpha 2 beta 3 gamma 2 and alpha 3 beta 3 gamma 2 subunit pattern. Other variants are also known. More than 19 variants are known actually, which may contain one unit from A1 to 6, B1 to 3, gamma 1 to 3, delta, iota, theta, and that way. So these dissimilar compositions will result in differential actions in various neuronal nuclei. The extraneuronal receptors, subtype A receptors, have usually alpha 4, alpha 6, and delta unit combinations. And the extraneuronal receptors have three characteristics. They have higher GABA affinity, less likely to exhibit desensitization upon continuous exposure of GABA, and third, they have ability to combine with general anesthetics such as propofol and ethanol. This is the depiction of the extraneuronal or extrasynaptic and synaptic GABA A receptors with different composition. Structurally, GABA A receptor chloride channel have binding sites for GABA, which are different from binding sites for benzodiazepine, zolpidem, and antagonist flumazenil. In addition, there are also binding sites for barbiturates and other substances which may act as agonist or antagonist. Now, benzodiazepines in particular bind at sites which include 2 alpha 1 subunit, 2 beta 2 subunit, 1 gamma 2 subunit, but they can also bind with the variants such as containing alpha 2, alpha 3, alpha 5 subunits in the different part of the brains. In contrast to this, Z drugs, that is zolpidem, zaleplon, zopiclone, S-zopiclone, they will be acting only on those types of the GABA A receptors which have alpha 1 subunits only, and this provides the hypnotic selectivity to Z drugs without having muscle relaxant or anti-epileptic action. GABA A receptor ionotropic chloride channel has three functional sites: the central chloride pore, the GABA binding site that binds with the interface of alpha and beta on two sides. So there are two GABA binding sites, whereas there are adjacent sites, allosteric sites we call as modulatory sites, where BDZ, benzodiazepines, bind at the interface of alpha 1 and gamma 2. Now, because of the variance in these subunit structures, differential binding has been noted. As I stated earlier, Z-drugs will bind only to alpha-1 subunit containing GABA. GABA B receptor, in contrast, is a metabotropic heterodimer receptor which belongs to class T of G protein couple receptors. Being heterodimer, it has two different subunits, beta 1 and B1 and the B2, and we'll see that B1 is a binding site and B2 is an action site, a functional site. It is located both presynaptic and postsynaptic membranes, whereas most of the GABA A receptors are located postsynaptically. The presynaptic GABA B receptor are autoregulatory, therefore regulate the release of GABA according to the need. In addition to this homotropic inhibition, it also shows heterotropic inhibition of the release of other neurotransmitters in other neuronal circuits. Now, postsynaptic receptors work by activating G protein and its downstream events. The binding of the GABA on GABA B receptors is at the B1 subunit in a venous fly trap manner, that is, the lid shuts when GABA binds, and this in turn activates the beta 2 subunit allosterically. Beta 2 subunit couples with GI and GQ. When it couples with GI-G protein, it inhibits voltage-gated calcium channel, activates potassium channels, and inhibits adenyl cyclase. Whereas when it combines with GQ, then it increases phospholipase C activity. All these ultimately will result in downwards protein kinase. Now, differentiating these two receptors further, there could be an agonist which directly binds to the receptor muscimol. There can be an antagonist bicocooline for GABA A. In a similar manner, there is an agonist baclofen which binds with GABA B, and there is an antagonist 2-hydroxycyclofen which is specific for GABA B receptors. In addition to this, there are allosteric modulators. The positive allosteric modulators for the for the GABA A receptors are benzodiazepines, barbiturates, ethanol, neurosteroids which are anesthetic, and volatile general anesthetic propofol. Whereas allosteric modulators will not be seen on GABA B receptors. Negative allosteric modulator have also been identified, although it is known as a pure antagonist, that is flumazenil for GABA A. In addition to this, chloride channel, the central chloride channel may directly be affected by barbiturates or blocked by picrotoxin. GABA A receptors have an important role in the motility disorder, that is the neuromuscular motor ataxia, in Huntington chorea, in Parkinsonism, epilepsy, sleep disorders, and drug addiction. Whereas the GABA B receptor mostly spinal, so it is involved in spastic disorders and to some extent sleep. Now these GABA and GABA receptors. GABA as a neurotransmitter interact with so many pathways in the brain because interneurons are present which interconnect with several axons and dendrons. In mesolimbic system, GABA increases dopamine release, thus reduces craving for drugs and craving for ethanol. And this is the main mechanism of action of sodium oxybate called as gamma hydroxybutyrate. Now this agent acts preferentially on synaptic GABA B receptors and also it acts on extrasynaptic GABA A receptors. GABAergic interneurons in the prefrontal cortex modulate presynaptic NMDA excitatory activity. Ketamine inhibits function of NMDA receptors on the GABAergic interneurons heterotopically and thus reduces neuronal excitability. Basal ganglion interaction is very typical The striatum is the receiving station of basal ganglia. It receives glutaminergic input from cortex and, more importantly, dopaminergic input from the substantia nigra, that is pars compacta part. Striatal output to globus pallidus interna and substantia nigra pars recta is direct by inhibitory GABA and indirect through globus pallidus externa, that too involves GABA. Globus pallidus externa in turn sends inhibitory GABAergic signals to subthalamic nuclei, and the subthalamic output is again excitatory glutaminergic to substantia nigra pars recta and globus pallidus interna. These two nuclei are GABAergic output producing, and that goes to thalamus. The thalamus uses glutaminergic transmission to motor cortex. Now in this setup, there is a close association activities between excitatory and inhibitory neurotransmitters, namely dopamine, GABA and glutamine. And any disturbance in one pathway can lead to disturbance in the motor and gait and abnormal movements appear. So this is an important to understand how these close. Now pathophysiological role is characteristically seen in Parkinson's disease, and primary defect is destruction of dopaminergic neurons in the substantia nigra pars compacta, and this neuronal activity will result in altered GABAergic neuronal activity in globus pallidus externa. And since it is not affected, it is only the dopamine neurons are destroyed. So therefore the persistent GABAergic activity through globus pallidus externa results in imbalanced stimulation of thalamic nuclei, which in turn stimulate motor cortex. So abnormal movements appear. Uncontrolled level, abnormal movements appear. Now converse is seen in Huntington chorea. There is critical reduction in the activity of the GABA synthesizing enzyme GAD, and this is an importantly seen in striatum and cerebral cortex. So there is a reduction in the GABA concentration or decreased activity of GABAergic neurons, and degeneration of the caudate nuclei begins from its tail, and there is unaffected dopaminergic activity. So again there is an imbalance. This imbalance because of reduced GABA, whereas in Parkinsonism it is because of reduced dopamine. In anxiety and mood disorders, reduced activity in GABAergic pathways is supposed to cause anxiety disorders, and it also has a role in genesis of depression and mania. Chronic alcoholism has been shown to have down-regulation of GABA A receptors, and that may be responsible for. Now, there are some uncommon inherited disorders where we can see change in the GABAergic neuronal activity. Mutations in the ligand-gated channel GABA A have a role in genesis of idiopathic epilepsy. Now, the enzyme GAD has two isoforms, GAD65 and GAD67, and they are involved in GABA synthesis. GAD67 deficiency due to inherited haplotypes is associated with isolated cleft lip in transgenic mice, and this is an uncommon manifestation. Now another enzyme deficiency involves GABA T and succinic semialdehyde dehydrogenase. Now these are rare. Now we will see that some abnormal movements occur due to deficiency of these enzymes, including succinic semialdehyde dehydrogenase deficiency manifests autosomal recessive gene at the chromosome locus 6p22. So the GABA is not converted to succinic acid but accumulates as the gamma hydroxybutyrate or GAB in blood, and large amount is excreted in the urine to the extent of 200 to 800 fold. The syndrome is characterized by hypotonia, ataxia, seizure, and neuropsychiatric manifestation, including sleep disorder. Elevated levels of the metabolite 4-hydroxybutyric acid indicates the defect, and there will be need for further. Another uncommon syndrome is GABA transferase deficiency syndrome, which is again a rare autosomal recessive caused by mutation in the gene ABAT transferase gene, and is characterized by recurrent seizures, choreoathetosis, hypotonia, and somnolence in children. Extremely uncommon is homocarnosinosis, and homocarnosine is a brain-specific dipeptide formed by GABA and histidine during the metabolic pathways of GABA. It is found in higher concentration in dentate and inferior olivary nuclei, substantia nigra, intermediate concentration globus pallidus, and lowest concentration prefrontal cortex, caudate nucleus, and nucleus accumbens. Now, this is characterized by early onset spastic paraplegia in too young. So, there are many drugs which act through GABAergic pathways. Many of them are known. The most prominent ones are anti-epileptic drug, tiagabin, which inhibits GAT enzyme, vigabatrin irreversibly inhibits GABA T enzyme in astrocytes and neurons, pregabalin, which is related to GABA, although it is structurally related, but the main mechanism is the inhibition of N type of calcium channels and inhibition of voltage-gated potassium channels. Only minor pathway is GABAergic inhibition. It is used in focal epilepsy, neuropathic pain, and anxiety. Gabapentin also has the main mechanism on the channels, but it also facilitates presynaptic GABA release. Uses are same as for pregabalin. Valproic has multiple mechanisms, including increased GAD activity and reduction in the conversion of GABA to succinate by inhibiting GABA T, just like vigabatrin. Stiripentol increases GABA release and inhibits presynaptic uptake of GABA, and that has also been used in childhood epilepsy. Benzodiazepines are well known as positive allosteric modulators A receptors. Among the other drugs which may act on the synaptic and extrasynaptic sites of the membrane include general anesthetics, etomidate, propofol, gamma hydroxybutyrate, which can also bind with GABA A subtypes having A4 and delta subunits. It is a weak partial agonist of GABA B receptor also. It has been tried in chronic alcoholism and narcolepsy, but unfortunately it is mostly misused by bodybuilders, allegedly increases growth hormone level. It has been misused as a date rape drug, and it has a great abuse potential, habit forming potential. Neurosteroids are the drugs which combine with the synaptic and extrasynaptic sites of the GABA A receptors having delta subunits, and they include alprazolam, brexanolone, and ganaxolone. Brexanolone, for example, is approved for postpartum depression. They all have anesthetic sedative property. Acamprosate is used in chronic alcoholism, although it is a weak NMDA antagonist, but has GABA A receptor activity. And finally, ivermectin, that is used in the treatment of nematodes and arthropods. It acts by intensifying GABA mediated inhibition in parasitic peripheral motor nerves, and this is how many of the drugs